Okay, we are going to finish everybody talking about our synapses. So really just run through the opposite examples where we left off last time. And then very briefly talk about some of the other cells that exist in our central nervous system. So you saw the announcement, but I cut down those slides substantially so that next time we can start off where we need to start off in our next unit, really focusing in on, okay, how do we send signals to other parts of the body and start to think about things like what really so that we don't overload our next unit. When we were last talking, we were talking about modulator neurons, essentially. So we were talking at the end about how we can sort of fine-tune the signals that our postsynaptic neurons are getting. So we talked about this idea of divergence. So we can have one neuron, a presynaptic neuron, sending out signals to many cells. We can have the opposite convergence, many different neurons sending a signal to one postsynaptic neuron. And that's modulation bit is we have some neuron separate kind of from this chain affecting how the presynaptic neuron is releasing the neurotransmitter at its synapse of the postsynaptic neuron. Right, so modulator neuron connects in an axo-axonic connection. So the axon here is connecting to the axon of the presynaptic neuron. The example we walked through is on the left here, the presynaptic facilitation. So when we see that word facilitation, one of the things like to facilitate something is to make it happen, right? So presynaptic facilitation is when we have more neurotransmitter being released due to this modulation. So here we see our modulator neuron E coming in, affecting presynaptic neuron C here. So D and C are converging on X. E is modulating the expression from C. Right? And we can see that when both C and N fire at the same time, we've got more neurotransmitter released, more neurotransmitter here. We're going to assume that this is a neurotransmitter that creates a depolarization, right? So it creates basically probably sodium moving into the cell, gets us closer to our action potential threshold. Okay. So we want to make sure that we understand the opposite. So less in the way. Right. So we can't have the opposite thing happen too. So we can facilitate, we can make our neuron, our presynaptic neuron release more neurotransmitter, or we can inhibit, we can make it release less neurotransmitter. So this is just exactly the same, but we're doing the opposite thing. All right. So in this situation, we see we have a presynaptic neuron G, and we have a presynaptic neuron F converging on Y. Right, so that's our sort of baseline setup here. And we have this modulator neuron H. Isn't touching Y, it's touching F. So that's that axo-axonic connection there. And so when H fires, it's going to affect what F is doing. So we can see under our normal connection conditions, when F fires, it's a little bit of a depolarization here. So that was one of our presynaptic neurons. If G fires, creates a little bit of depolarization as well. If H fires, H isn't synapsing on Y, so we wouldn't expect to see any change at all to Y, right? When H fires. If F and H fire together, This is what we're trying to understand with the inhibition bit, right? So under our normal conditions, it's just F. F creates depolarization, so it creates an excitatory postsynaptic potential. But if H is going at the same time, nothing, right? So this is because H is inhibiting the action of F. So when these two fire together, this means that we have much less neurotransmitter release. So in this first example, this arrow is showing you just F firing. In this second picture, F is firing at the same time as H, and H is inhibiting F. So think it's like creating some sort of hyperpolarization kind of in F, right? So that would be kind of the mechanism happening here. All right, so we'd be lowering that membrane potential. The membrane potential is part of what drives calcium flooding into the cell. All right, so if these two fire at once, less neurotransmitter, less depolarization. And we can see just fully putting out our combinations, F and G are each working independently. They each created depolarizations because graded potential sum, if they fire at once, we have more of a depolarization, its threshold, fires off our action potential. Right? If F, G, and H all fire together, what we're going to add together is G just acts on its own, right? Nothing else is touching it. So we'll add this depolarization. But when we see F and H both firing, we know that we need to look at this modulated effect. So we're going to add it together with that F plus H firing, which is why we don't see this hitting threshold when all three fire at once. So questions at this point? No time for questions throughout. We're going to do an example. So we're imagining a sequence where we have neuron 1, which is a presynaptic neuron, and then neuron 2, our postsynaptic neuron. And then we're going to think about the action of a modulator neuron. So modulator neuron X has an axo-axonic connection to neuron 1. It's going to increase the amount of neurotransmitter that neuron 1 releases. So which of these is best describes this situation? Like, what is happening here? What am I describing? A couple seconds. Okay. So the correct answer is presynaptic facilitation. So it's worth just taking a look at it, that we're all on the same page with the difference between presynaptic facilitation and our excitatory postsynaptic potentials. So when we're talking about those two things, they do seem very similar, right? So an excitatory postsynaptic potential, do that one first. My neuron. And here's my postsynaptic neuron. Whatever it looks like. Okay. So an excitatory postsynaptic potential is telling you about what's happening with the voltage in our postsynaptic cell. So to be excitatory, an excitatory postsynaptic potential, we'd want to see that voltage go up, right? So our little graph would have a little bump, right, for our excitatory postsynaptic potential. This is going to happen probably because a neurotransmitter has ultimately allowed sodium into the cell. Could also happen if we close a channel that was letting potassium out, but anything that keeps that charge up from -70 is going to create excitation. Excitation because we're more likely to hit threshold and fire off an action potential. For presynaptic facilitation, that's when we're thinking about neurotransmitters. So when we're thinking about, when we're thinking about the release of the neurotransmitter from the presynaptic cell. So there we're thinking about modulation over here on our presynaptic neuron. So we have that third neuron there, that neuron X. Okay. And what this is describing is that when neuron X releases its neurotransmitter, it affects the amount of neurotransmitter being released by neuron 1, that presynaptic. Okay. So the facilitation still means like more, but instead of talking about more voltage in the postsynaptic cell, we're talking about more neurotransmitter from the presynaptic cell. This is just a redefinition of those connections. So modulator stuff, we're talking about those axo-axonic connections. When we hit axon to axon, axon terminal to axon terminal, we're only going to affect one synapse because we pass all the other parts of that cell. So we're not going to affect the soma of that cell. We're not going to affect the dendrites of that cell. We're just affecting that one axon terminal that we're touching. So it's really only going to affect one synapse, either ultimately exciting or inhibiting, selective, versus the axodendritic and axosomatic connections. If you have your neuron this, right? If you have a connection here, right, whatever happens at that dendrite and the body of the cell now affects the whole neuron, right? So that would then propagate down the axon. And if we have multiple axon terminals, right, so like spread them out. Sorry, I only drew one. Okay. It's now going to have an effect on all of those axon terminals because we just have that one axon coming up. So we go here, go here, go here. So this is not synapse, it's affecting all of the synapses from that neuron sequence. And this is just another diagram for you to practice on. There's nothing new here. We just have a different picture of a neuron than before and another kind of flow chart about what's happening in that presynaptic neuron versus what's happening in the postsynaptic neuron. We're going to move on and talk very briefly about the central nervous system so that we can spend all our time in the peripheral nervous system moving forward. So you've started to look at the central nervous system in lab. So your central nervous system, should not be a surprise, is made up of your brain and your spinal cord. Your brain and your spinal cord here at the top are responsible for information processing. Right? They're necessary for maintaining homeostasis. So we saw that central nervous system way back at the beginning of our first unit when we talked about homeostasis, right? That was the part of our nervous system that's receiving signals from receptors in response to stimuli and planning out our response. So planning, are we doing negative feedback here? Which direction do we need to go? What effector organs need to be affected? So this consists of 100 billion neurons and 100 trillion synapses. So this is a really complicated process in terms of how much energy it's taking, how many cells are devoted to this job. Okay. Other parts of the nervous system, so what we will be talking about in the future, are our nerves coming in and out. So our peripheral nervous system, we have sensory nerves that come in. Those are referred to as afferent nerves. Those would be artificial, so they're coming into the central nervous system. Also part of the peripheral nervous system would be our efferent nerves. So efferent nerves, we also refer to as motor nerves. Those are the ones going out. Okay. So when we talk about the nervous system, we really focus on the nerves themselves. We focus on the neurons. There are other cells in this system. So neurons are the cells that have electrical activity, so conduct impulses, but they generally can't divide and reproduce, or when they do, it's hard. You see it sometimes, but rarely. So usually when your nervous system is adapting to things, or you think about regenerating brain function after surgery, we're usually thinking about altering the function of neurons that exist. So we're thinking about changing synapses, changing connections, rather than generating new neurons. Although that is an area of ongoing research. Okay. The cells that we want to just know for today exist are the other cells. So our glial cells, or our neuroglia, support those neurons, but they can divide, and they're not really electrically active. So they're just kind of surrounding these nerve cells. One of the condensing pieces is that we have now on this slide just all of our types of glial cells. So if it should be a trick, you should probably expect there to be one of these glial cells somewhere on the exam, just asking you which of them it is. Right? So we got 5 types. Right? We have our astrocytes. So our astrocytes, their major function, although they have many functions, is to create something called the blood-brain barrier. So this basically helps protect your brain by controlling what is able to get from your bloodstream into that nervous tissue and the area surrounding it. So you can see this astrocyte here. It's covering over some gaps in our capillary here. So we got a blood vessel, astrocyte with these little foot processes kind of covering over the edges of that capillary, allowing us to kind of control what's traveling across the wall of the blood vessel. Our ependymal cells line in cavities. So when you look in lab at the ventricles in the brain, those empty spaces, they're not empty, they have cerebrospinal fluid, so they have liquid inside them. The ependymal cells are the ones that are making that. Okay. So here we see an ependymal cell next to a ventricle that would be producing that cerebrospinal fluid. Microglia are they have immune function, basically. So here we see a microglial cell. So they're going to protect that central nervous system from foreign matter by eating it, essentially. So phagocytosis is taking in one of these pathogens or foreign matter or dead matter and decomposing it, breaking it down, basically. So that's what the microglia do. And then we have 2 types of cells that make myelin. We have our oligodendrocytes and our Schwann cells. Okay? And their job is to make myelin. So remember, myelin, we talked about when we talked about propagation of the action potential. So when we talked about the axon and how it spreads the charge. Look at this. Multiple axon terminals here. Okay. So the myelin, we said, usually in an unmyelinated neuron, when we don't have myelin, we have to kind of activate each little patch of membrane separately in order to send our action potential down the line. It takes a bunch of time, relatively, because we have to trigger all the little points on that axon. Myelin is like insulation. So myelin wraps around that axon of a neuron and it speeds stuff up. So it speeds stuff up because now we don't have to trigger every patch of membrane. We jump through a process called saltatory conduction from node to node. So between our nodes of Ranvier, so we can have faster connection in our nervous system. So our action potential moving faster down the axon, so down this part of the neuron, towards our next neuron that we're going to synapse with. So the difference between the oligodendrocytes and the Schwann cells is just where they're making that myelin. Okay? So otherwise, you can think of them as the same thing. So oligodendrocytes are in the central nervous system. So they make myelin, but you'd see them in the brain or spinal cord. Okay? So here we can see an oligodendrocyte. And we'll also notice that the oligodendrocytes are forming myelin on multiple neurons. So spreading out. Okay. Versus our Schwann cells or myelin in the peripheral nervous system. So like you have a myelinated nerve out in your arm, right? That would be made by a Schwann cell. And you can see those Schwann cells just wrapping in circles around just one individual neuron, specifically around one individual section of a neuron. That is the full list of our glial cells. Right? So if you have to mention them, just because there are so many of them in terms of the number of cells in your nervous system, like 70 to 90% of your central nervous system isn't actually neurons, it's neuroglia. So those are functional support for the central nervous system. We also got physical support for the central nervous system. Right? So we've got our bony structure, the skull, protecting you from like impact. We've got our meninges with their layers, the dura, the arachnoid, the pia mater, helping anchor your brain and your spinal cord in place to that bony support structure. On the inside of the ventricles and then spreading in the subarachnoid space, so spreading between layers of your meninges. We have cerebrospinal fluid, so this is kind of more cushioning, and it is a watery fluid that kind of bathes the CNS and helps feed it and protect it, help that environment be kind of more controlled. The composition of the fluid in your cerebrospinal fluid is a bit different than the composition of the plasma running through your circulatory system. So we're creating kind of like a different external environment for those neurons and those glial cells. So that CSF is coming from the ependymal cells, circulating through the ventricles to the subarachnoid space. It eventually goes back into your veins, so it gets reabsorbed by arachnoid villi, which are just projections of the arachnoid. So basically, you want to know, it exists, it's important, it's in ventricles, and it's made by ependymal cells. This is the level we kind of want to be at. So we're cushioning the brain physically and maintaining that stable interstitial fluid environment for our cells in our central nervous system. Okay. So next, blood supply. Other than just knowing the very basics of what the glial cells are, I think this is the other most important topic when we're thinking about the central nervous system, this blood-brain barrier and this blood supply to the central nervous system. Because this is a place that you see potentially problems when things go wrong. So your central nervous system is about 3 to 4 pounds, so about 2% of your body weight, but it takes a really disproportionate amount of your blood supply. So it's getting 15% of your blood supply, even though it's only 2% of your body. Right? This is because your brain really needs a lot of energy to function. Okay? It has a high metabolic rate, so we're converting lots of glucose to ATP to help with that cellular function. And this means that we need lots of oxygen. So when you are at rest, your brain is using 20% of the oxygen in your bloodstream, right? So 20% of the oxygen, 2% of your body weight. Your brain is actually using 50% of the glucose that is consumed by your body at rest. So if you're studying, thinking really hard and getting hungry, right? That's kind of real. Your brain is really using all that energy. It's really using that much glucose. So the brain really depends on blood flow, right? It's got a lot of blood flow, and that blood flow is super important to it. If you don't get blood to the brain, you really don't get any blood to the brain, you are going to die, right? Your brain doesn't really have a backup system at all, right? And it's critical to your function. So needs lots of blood because it's doing a lot of stuff, high metabolic rate. The other thing that happens to be true about your brain is that it can't really use anything other than glucose for energy. So other cells in your body can break down glycogen potentially. It can potentially break down fats, triglycerides, maybe they can break down proteins, right? Depending on your nutritional state and what cell we're talking about, but your neurons can't do that. So they really thrive on glucose and practically nothing else. There's almost no glycogen storage in your central nervous system. Tiny bit in some glia. Can't use fatty acids for energy. Under really extreme conditions, you can use a ketone or two, but it really wants glucose. So hammering that home, because when we talk about the endocrine system and glands and hormones and digestion, it's going to be important to know that the brain needs all the glucose it can get. When we have disruptions to the blood supply to the brain, this is a problem. That's what a stroke is. So when you have a stroke, what you're really doing is saying that you have damage of some type to a blood vessel in the brain, which is part of what's causing those changes in behavior and function. So this can happen either because a blood vessel gets blocked. That's what occlusion means. When something gets blocked, something's occluded, it's cut off. Hemorrhage can also create a stroke. So hemorrhagic strokes are a thing. So if you have a blood vessel that bursts and you're bleeding in your brain, is not really a good way for those cells to get their tasty glucose and oxygen. So that's also going to create regions of the brain that are not getting enough blood. So we got that blood-brain barrier. Mentioned it when we looked at those astrocytes. Okay, so we do need blood to the brain. It's coming up through arteries in your neck and your vertebrae. Arteries get smaller and smaller. Our site of exchange is in the capillaries. So that's like the smallest branching of your cardiovascular system right before you go to a vein. Capillary walls are pretty thin. So capillary walls are really just a layer of endothelial cells. So they're like one layer thick here. Okay. And they have junctions between them. Okay. So they have junctions between them where some stuff can get out. So here you can see pores between those endothelial cells. And we can see that label says that some hydrophilic solutes are able to travel through those pores. Okay. We don't want that to happen in the brain. We want to really control everything. Okay. So we are going to join everything tighter. So those capillary walls are going to look different. We got tight junctions here. And we've got those astrocytes kind of modulating all of this, holding those capillaries tighter together and really limiting the amount of exchange. So you can see how they're like even blocking parts of the membrane, right? So whereas here we had some transcytosis happening, you've got an astrocyte over that patch. It's got to go through the astrocyte if it wants to get anywhere. So this is important when you think about like what medications could get into your brain, what type of pathogens could get into your brain, right? Knowing that we have this separation between the blood and the brain in specific that's different than in the rest of the body is important to eventually understanding those things. Finally, this is just zooming in on that myelin again, right? So this is expanded, but really the basics is what I said on that previous slide, right? Myelin wraps around the axons of neurons, speeds up conduction, so speeds up how fast that action potential travels. It's formed by two different types of glial cells, depending where you are. Okay. So here we're just looking at that myelin sheath in the peripheral nervous system. So instead of my drawing, you got an actual drawing here. So you can see those nodes of Ranvier labeled out for you. You can see the axon in the center in blue, and you can see these Schwann cells wrapping around. Okay. So there is a technical name for that structure, that wrapping around of the Schwann cell, called a neurolemma. Okay. It's characteristic of the peripheral nervous system and these Schwann cells. We don't see it in the central nervous system where the oligodendrocytes are making them. So, gaps, nodes of Ranvier, speeding stuff up. Again, just another summary slide, like zooming in on that. Okay, so we got going on in the central nervous system versus what's going on in the peripheral nervous system. So central nervous system, we've got a different type of glial cell, the oligodendrocyte. Now that I'm looking at this word, if you think dendrites, you think dendrocytes, so think about those processes on the bodies of our nerves. Maybe that can help you remember that it's in the central nervous system. And the one oligodendrocyte can go out to many neurons. Okay. Still makes myelin, but this neurolemma name is specific to the structure you see in the peripheral nervous system. Peripheral nervous system, don't got a trick for you. These are just Schwann cells wrapping around forming this package. And fun fact about this is that when you talk about like white matter, gray matter, if you talk about the brain, what it's doing, what different parts of the brain are doing, that white color, when you have white matter, this is coming from that. So the myelin is what is white in the white matter. So if you have white matter, we're talking about axons. We're talking about those connections in the brain. Right? The gray matter is made up of stuff that is not covered by myelin. So usually we're thinking about like cell bodies, dendrites, and some small neurons where we have axons that are not covered by myelin sheaths. So gray matter you can see in the central nervous system, because really in the peripheral nervous system, you have to go far, so you want to get there fast. So pretty much everything is myelin. And that is it. Okay. So that's words on the slide, but really just expect give me a description of a type of glial cell, and you will tell me which one, and then I will ask you some questions about blood. That's what we want from here. So we are now going to move into a review. Okay. Have a quick list of topics so I can rapid fire in the next 15 minutes, try to talk through this whole unit, or you can ask me specifically what you would like to go over. So I'm going to give you a second, see if anybody has a most important topic that they want to talk about. Okay, cool, can answer these quick. So when you're trying to figure out presynaptic facilitation or presynaptic inhibition, all you're trying to figure out is, are you releasing more neurotransmitter? Are you releasing less neurotransmitter? So if there's more, it's facilitation. If there's less, it's inhibition. And it doesn't actually matter what that neurotransmitter is or what it does. It's just about how much neurotransmitter is released. So that's a point at which you don't really have to think about excitation at all, or that postsynaptic potential at all. It's a kind of separate. Okay, switch into this hover cam then. I'll keep checking for questions, but otherwise I will just rapid fire go through the little list that we've covered. Sorry, I don't want to write upside down for y'all, but also drawing receptors upside down is going to be a bad time. Someone tell me what I clicked on that got me that rotate button. Because I remember. You. Okay, cool. Now we're right. All right. So when we started out, we were talking about how we set up a membrane potential for our neurons, that resting membrane potential. And we focused on equilibrium potentials and how they affect that. Okay, so we started with our little body of a basic cell. So we want to think about this as really the soma of our neuron, but this is happening throughout. Okay? And we know, based on our equilibrium potentials, that sodium wants to go into the cell. Right? Sodium wants to go into the cell. That is based on its electrochemical driving force. Okay? And sodium wants to go into the cell because both its electrical driving force and its chemical driving force are pointing in. The electrical driving force, because that positive charge on the sodium is initially attracted to the negative charge inside the cell. The chemical driving force, because of the action of our sodium-potassium pump, that sodium-potassium pump uses ATP, so active transport, primary active, to pump 3 sodium out, okay, and pull 2 potassium in, okay, which is creating a high concentration of sodium outside the cell versus low inside, and creating a high concentration of potassium inside the cell versus lower outside. These labels of high are labeling where we have high sodium, where we have high potassium. Opposite side of that cell membrane is where you would see the low side. Chemical driving force always goes from high concentration to low concentration. So that's why it points in for sodium. So for potassium, same thing, right? So now we gotta do the same thing, but opposite direction. So we go from high potassium inside the cell to low potassium outside the cell for our chemical driving force. Our electrical driving force, this is still just an attraction between the charge on the ion and the charge inside the cell. So our electrical driving force, we've got that positive charge on potassium attracted to the negative charge inside our cell. Okay. When we add this all together, because of these relative sizes of these two forces, potassium has an equilibrium potential that is more negative than our resting membrane potential. So our electrochemical driving force for potassium points out. So that's how we set up the forces on each of these ions. When we're thinking about the neuron as a whole, okay, the reason we are at -70 is that we have a push and pull going on between sodium and potassium. Sodium's equilibrium potential is up at about +60. The equilibrium potential of potassium is like -90 something. Okay. But we have a lot more channels, a lot more leak channels for potassium, or in other words, our membrane is more permeable to potassium. So because we have a lot more channels for potassium, more potassium is getting out of the cell. And it's not being canceled out by the amount of sodium that gets in. So even though we have a strong force for sodium to move into the cell, we have 25 times more permeability to potassium, which is why our equilibrium potential, our resting membrane potential is close to that equilibrium potential for potassium. So that's why we start out there at rest. The next thing we talk about, we're leading up to how we get to signaling, right? So signaling to another neuron, signaling to another cell. Okay, so the key part of this is the action potential. We talked about graded potentials first, but we're going to lump those in now that we know more details about our excitatory and inhibitory postsynaptic potentials. So for our action potential, what we're looking at is a change in the resting membrane potential. All right, so we're going from -70. We're eventually going to get up to +30. And along the way, we have to hit a threshold value of about -55 if we want to fire off an action. So what happens to that voltage inside the cell? So we start out at rest. We'd have some graded potentials adding up together until we get to -55. Then we see a spike up to +30. This is our depolarization. Okay. So the depolarization phase. Depolarization. Then we see repolarization, so coming back down. This is repolarization. Overshoot a little bit, so we go down below resting membrane potential, so that's hyperpolarization. And then eventually we get back to rest at some point. This is all driven by our ions traveling, right? So the reason we care about the threshold, right, is that at this threshold, what we have happening is that our voltage gated sodium channels react. Okay, so voltage gated sodium, okay, and specifically our activation gate opens first. So an activation gate. Okay, we know sodium wants to get into cells, so it does, right, which is why we see that depolarization phase. It stops because at this point we see the inactivation gate closed. So this is still our voltage gated sodium channel functioning. That voltage gated sodium channel Right now, what's happening is that inactivation gate is closing, so blocking over that pathway that has two doors. So sodium is no longer getting into the cell. And the other thing that's happening at the same time is that our voltage gated potassium channels are kind of slow. Sorry, I should have brought some colored pencils. Voltage gated potassium channel. This has one gate, so we don't have to name it. But that voltage gated potassium channel opens. We just talked about why potassium wants to leave cells. So potassium leaves the cell, which is what's causing that repolarization and that hyperpolarization until eventually both of these reset to their original state. So our potassium voltage gated channel is closed, and so is the sodium voltage gated channel because its activation gate is closed, inactivation gate has opened back up. During this process, we also have a refractory period, right? So refractory periods have to do with when it is difficult or impossible to fire off an action potential. I'll redraw it because that's getting kind of dense, right? So same old action potential, every action potential looks exactly the same. That's why we have that all or none principle. We either have one or we don't. So during our absolute refractory period, we absolutely cannot fire off another action potential. So the reason we can't fire off another action potential is that those voltage gated sodium channels have been inactivated, right? So we have to wait for them to open back up. So that's really going to happen like during the action potential itself, pretty much, right? We might draw this line a little bit over because the voltage gated channels are not all doing the exact same thing on a flick switch at exactly the same time. So when they're resetting, sometimes during the end of this phase, we've still got a bit of a hyperpolarization. So we're a bit low. So we'd have to have a lot more sodium come in to make up for that. So that's the relative refractory period. Because we're probably not going to have another action potential, but if we have a strong enough impulse, we could. Refractory versus absolute refractory. We'll flip over for our synapse. All we're talking about is the connection between our two neurons, our presynaptic and postsynaptic. Our presynaptic neuron, when we have that action potential in, releases neurotransmitter because it's in vesicles that end up in the synaptic cleft. This is because calcium floods into the cell because it also has a voltage gated channel. Okay. On the opposite side, on that postsynaptic neuron, we've got receptors. Neurotransmitters bind onto receptors, which ultimately open a channel, allowing or open or close a channel, allowing more ions to flow or preventing ions from flowing. We've got two types of receptors. We've got the channel linked or ionotropic receptors. These are just ligand gated. So literally just that neurotransmitter binding opens or closes it. And we've got our metabotropic receptors. Oh, it's not awesome. All right, these ones are more complicated. We still have a neurotransmitter bind on. But then we got this like three-piece G protein that breaks apart, goes and affects other membrane proteins, sets off some second messengers, potentially. Right, but ultimately, those second messengers are going to open or close an ion channel. Right. So ions are either going to flow in, oops, sorry, flow in or flow out, depending on what that channel is, depending on what that neurotransmitter was. Okay. If we end up with more positive charges inside the cell. Okay. So more positives inside, that's depolarization, which is excitatory. So an excitatory postsynaptic potential. That's right, excitatory, that should be nice. Excitatory postsynaptic potential. Okay. Or less negative, right? If we have less positive charges ending up or more negative, we have an inhibitory postsynaptic potential. And all this has to do with is, are we more likely to fire off an action potential or not? So this has gotten us with the excitatory postsynaptic potential up closer to that threshold value, in which case we fire off. The inhibitory got us down below, harder to fire an action potential. Sorry, we're 3 minutes over, so that pretty much gets us to where we started today. If you have questions, I'm happy to answer them. So I'll stick around for a minute.
It's always do that every coach after every like at every class, it's 3 minutes.